Welcome to today's lecture on histology. Today, we will be exploring the fascinating world of ocular histology, delving into the intricate microanatomy of the eye and understanding how various structures within the eye contribute to vision. Let's dive right in. The cornea is the clear outer structure of the eye. It consists of 5 layers: epithelium, outer layer, Bowman's layer, thin layer of collagen fibers, stroma, thickest layer of collagen fibers and cells, Descemet's membrane, thin layer acting as a scaffold, and the corneal endothelium, innermost layer regulating fluid transport. Knowledge of corneal microanatomy aids in diagnosing and treating corneal disorders. The pupillary reflex regulates the size of the pupil in response to changes in light. This reflex is controlled by the sphincter pupillae and dilator pupillae muscles, which constrict or dilate the pupil respectively. It is influenced by the parasympathetic and sympathetic nervous systems. Abnormal pupillary reflexes can indicate neurological or eye diseases. Accommodation is the eye's ability to focus on objects at different distances. This involves the contraction and relaxation of the ciliary muscle and the shape change of the lens. Age-related decline can cause presbyopia. Understanding the microanatomy of accommodation is important for managing refractive errors. The retina, located at the back of the eye, contains specialized cells called photoreceptors that are critical for vision. Rods are responsible for black and white vision and function best in dim lighting conditions, while cones are responsible for color vision and function best in bright lighting conditions. These cells contain photopigments that absorb light and convert it into electrical signals that are transmitted to the brain. The structure of these cells includes outer segments with photopigments, inner segments with organelles for energy production, and synaptic terminals for signaling. Understanding this structure is important for understanding vision disorders and developing treatments. The retina is composed of multiple layers, including the inner limiting membrane, ganglion cell layer, inner plexiform layer, inner nuclear layer, outer plexiform layer, outer nuclear layer, external limiting membrane, photoreceptor layer, retinal pigment epithelium, and Bruch's membrane. Each layer has a specific purpose in the visual process, such as transmitting signals to the brain and supporting photoreceptor function. Knowledge of these layers is essential for diagnosing and treating retinal diseases. The macula is a region in the retina that helps with central vision, detail perception, and color vision. At the center of the macula is the fovea, which has a high concentration of cone photoreceptors and provides sharp vision. The macula and fovea have specific anatomical features that optimize light transmission to the photoreceptors. Understanding these details is important for diagnosing and treating conditions like macular degeneration and macular holes. The choroid is a highly vascular layer located between the retina and the sclera. It supplies oxygen and nutrients to the outer layers of the retina. The choroidal vasculature consists of a dense network of blood vessels, including arteries, veins, and capillaries. These blood vessels help maintain the metabolic needs of the retinal pigment epithelium, RPE, and the photoreceptors. The choroidal vasculature also plays a role in regulating temperature and removing waste products from the outer retina. Understanding the microanatomy of the choroid is crucial in diagnosing and managing conditions such as choroidal neovascularization and central serous chorioretinopathy. The retinal pigment epithelium, RPE, is a layer of cells that supports and protects the photoreceptors in the eye. It absorbs light, maintains the blood-retinal barrier, and helps remove waste. Understanding its structure and function is important for diagnosing and treating eye disorders. The optic nerve pathway is responsible for transmitting visual information from the retina to the brain. This complex pathway begins with the axons of ganglion cells, which converge to form the optic nerve. The optic nerve then travels towards the brain, crossing over at the optic chiasm, leading to further visual processing in the visual cortex. Understanding the microanatomy of the optic nerve pathway, including the arrangement of axons, the presence of glial cells, and the regions of decussation, is essential in diagnosing and managing conditions affecting visual transmission, such as optic neuritis and optic glioma. Visual field defects are areas of reduced or absent vision within the visual field. They result from damage to specific parts of the visual pathways such as the retina, optic nerve, and visual cortex. These defects can show as scotomas, localized reduced vision, or hemianopias, loss of vision in half of the visual field. Causes include glaucoma, stroke, and tumors. The development of the retina is an intricate process involving the differentiation and migration of various cell types. During embryogenesis, retinal progenitor cells give rise to different retinal layers, including the ganglion cell layer, inner nuclear layer, and outer nuclear layer. Specialized radial glial cells guide the migration of developing neurons to their appropriate positions within the retina. Moreover, the outgrowth of retinal ganglion cell axons forms synapses with target neurons in the brain. Understanding the intricate processes involved in retinal development is essential for comprehending congenital retinal disorders and exploring potential therapeutic interventions. The eye relies on a constant supply of oxygen and nutrients to maintain its cellular functions. Ocular blood supply is provided by a complex network of blood vessels, including the ophthalmic artery, central retinal artery, and ciliary arteries. These vessels supply oxygenated blood to the various structures within the eye, including the retina, choroid, and optic nerve. Understanding the microanatomy of ocular blood supply, including the branching patterns, anastomoses, and autoregulatory mechanisms, is crucial in diagnosing and managing conditions such as retinal artery occlusion and diabetic retinopathy. Ocular histology allows us to comprehend the intricate microanatomy and functional organization of the eye. By understanding the complex details of the eye structure, healthcare professionals can improve their ability to diagnose and treat various ocular disorders. I hope this lecture has inspired you to explore ocular histology further.